Hi everyone, it's Dr. Snexer, and today we're going to be talking about the scientific method, which surely is something that you have thought about or talked about before in other classes, but I just want to give some examples and talk about how it's relevant to this course. As we have said already, there are a lot of goals that we have for this semester. Ultimately, we are trying to develop research proposals so that each of us can make an experiment, design an experiment, and talk about that, about how we're going to do it, about the different questions we want to answer. And so all of the activities and all the topics that we're talking about this semester are getting us to that goal. We've already focused a bit on the primary literature. Obviously, we've spent a few weeks talking about it, and that's because it's so very important. Not only is it the end goal of all scientific work to share our finished product with others, other scientists, everyone within the world, but the primary literature is also where we're spending a lot of our time so that we can develop our research questions and hypotheses. So if we take a look at this process of the scientific method, it really is more than that poster that's on the wall or those steps that you're forced to memorize for some high school biology class. The scientific method is the process that all scientists are using to really get at the answers. We want to know how the natural world works, and this process allows us to get those answers. Of course, our main focus so far has been sort of the end of this process, which is how scientists report their results within the primary literature and those scientific journals. But that's also actually the beginning of this process too, is doing background research, using those primary literature articles, those manuscripts that other scientists have already written about the work they've already done, so that we can get an understanding of what we already know and what we don't know yet, what we can actually still figure out. So this process is really somewhat circular. Not just as individual scientists, but as scientists as a whole, we're constantly sharing our work, and others are reading the work to understand sort of where their ideas fit within this process. So let's spend a little bit of time talking about this process. And I like to really spend some time thinking about science itself. We are all scientists in this course. And I like to think that we are all scientists on this planet. Carl Sagan, who was a famous astrophysicist, he wrote a lot of books, and one of his famous quotes is, "Every kid starts out as a natural-born scientist, and then we beat it out of them. Few trickle through the system with their wonder and enthusiasm for science intact." I know, kind of a downer, but I think it's an interesting quote to think about when we're thinking about science as a whole. Maybe not any of you in this course, but I see this when I talk to non-scientists, those who identify as non-scientists, who think science is hard, science is scary, science is difficult. It's only for those really smart, nerdy people. But I don't think that's true at all. I feel like we all have this natural spirit of inquiry. We want to know why things happen the way they do, how these things work. But maybe throughout the education system, starting from, when we're younger, we're often thinking about science as, a difficult subject. We have to memorize all the stuff that we already know about science. And so that often turns people off, right? Like Carl Sagan said, we beat out of young students the desire to know more because they're faced with this, test-taking mentality of learning things for the test rather than understanding this process of science, how it really is just an inquiry-based system that is both objective and valuing evidence to discover the truth. The truth is out there, and scientists are just trying to figure it out. So, what is science in our thinking of how everyone really is a scientist at heart? Well, science, if you're a word nerd like me, actually comes from the Latin root scientia, which means to know or knowledge. So science is knowledge. It's the body of knowledge about the natural world. So it is all that stuff that you had to memorize for the test. It is all the stuff that's inside your biology textbook or your chemistry textbook or your physics textbook. Science is that body of knowledge of things that we already know about the natural world. But science is also the evidence-based process for acquiring all of that knowledge. So I think about science as both a noun and a verb, right? Science is that information, right? It's a noun. It's the information that we already have about the world. But it's also a verb. It's an act of doing. It is a process by which we figure out all of that knowledge. I mean, I wouldn't recommend that you tell your friends you're going off sciencing today. So maybe not a verb in that sense, but it is a process, not just facts. It's the process by which we get those facts. And that's really the focus of this class. Using the scientific process to answer questions that we have. Questions always relate to hypotheses. Hypotheses are sort of these logical ideas that we get from inquiry. Inquiry is searching for information and explanation, right? Scientific inquiry is trying to think about the truth of the natural world. And the process of science allows us to take what we know and figure out more, forming logical hypotheses and conducting observations or experiments to support or reject those hypotheses. Observations are really at the core of inquiry-based science. Observations are really just recordings of what we experience within the natural world. I assume most of you in here have heard of one of the most famous observational animal behaviorists out there, shown here in this picture, Jane Goodall. Jane Goodall is a wonderful scientist who, back in the 1970s, traveled to Africa in order to study the chimpanzees in Tanzania. You can see a picture of her here as she's observing them. Like I said, I'm sure most of you know her story. There are a number of documentaries. If you don't, I definitely recommend you checking them out or reading some of her books. But the point I want to make about Dr. Goodall right now is that she was an observational scientist. She wrote down every detail of what she observed as she traveled throughout the reserves looking at these chimpanzees. And you can see her notebook here. She wrote down who interacted with whom. She named all of the different animals that she encountered, which was not typical of scientists at the time, but she was not your typical scientist when she went out, which is why she was hired in the first place. But she is a great example to look at because she really took this inquisitive approach to collecting data. and data is at the core of observations. We are going to spend the next couple of weeks talking about data, variables, hypotheses. But our first look at data here is to just really look at the definition and the types of data we might be able to collect. Data are just the recorded observations or items of information. We very broadly categorize data as either qualitative or quantitative. Qualitative refers to descriptive pieces of information. So maybe a description of who is near whom, which chimpanzee is standing near which other chimpanzee. Also the quality of the light, the characteristics associated with the environment that we're currently in. There's a stream that is stagnant. There is many large trees around the stream. So really just descriptions of the qualities of the situation that we're looking at, the observations that we're making. Quantitative data really has to do with quantity measurements. Quantitative data is typified by being able to be organized into tables and graphs, but it really just means counts, percentages, numbers. Now, very often, scientists turn long-term qualitative data into quantitative data because quantitative data can be analyzed. Again, this will be our topic for many weeks coming up, but analysis of quantitative data allows for mathematical models that can tell us if populations of data are similar or different, which is a little bit more difficult to do with qualitative data. But both of these types of data are important to collect and use in order to understand more about whatever it is that we're being inquisitive about, whether it's groups of chimpanzees or populations of endangered fish that live in the desert or pandas that are in the zoo, whatever it is that we're thinking about. We should be trying to collect data from the observations that we're making. From the observations and the data that we collect, along with the primary literature that we already have used for our research, we can propose hypotheses. We can think about research questions that don't yet have answers that we would like the answers to. So the basis of this is, of course, a hypothesis, and we often focus on hypothesis-driven research. Now, I assume everyone has heard that term hypothesis before. Think to yourself for a second. Jot this down in your notes. If I asked you right now, what is a hypothesis? What would you say? Go ahead, write it down. Did you write it's an educated guess? Is that what you wrote? Am I creeping you out right now? Because that's exactly what you wrote, educated guess. About 90% of the time, if I ask a student to define hypothesis, they will say educated guess. On the Regents exam, if you're a New York student and you took Regents biology, the answer to that question is a hypothesis is an educated guess. Personally, I don't like that definition. It's not incorrect, but it focuses on guess a little too much for me. Like, I know it has educated in front of it, so that's supposed to make it sound like you did your research before you made a guess. But very intentionally, hypotheses are not guesses. You know, I can guess at a lot of things. I'm guessing that the Buffalo Bills might win next week, their football game. Now, did I do any research in order to figure that out? No, I did not. I'm just guessing. I don't even know who they're playing next week. But I can throw a guess out there because, they may have won in the past and maybe they'll win. There's a 50% chance, right? No, actually, that's not true at all. There's not a 50% chance that they will win and the other team will lose. Based on information, data that we can collect, we can make a hypothesis as to who would win a football game because some players are better than other players. Certain conditions are better to play in than others. The Buffalo team is always good at playing in cold weather and maybe not as great in warmer weather, while other teams, their opponents, may be better in warmer weather versus colder weather. So there are a lot of variables. There's a lot of data that can be collected in order to make a better hypothesis than just a guess. And that's sort of my point when it comes to hypotheses. They are well thought out, tentative answers to well-framed questions. Hypotheses are provisional explanations of natural phenomenon. Basically, a hypothesis is explaining why the thing I observed happened. So I've made observations, I've done my research, and now I'm going to try to explain why or how those things happened. These well-framed questions, these provisional explanations, lead scientists to predictions that can be tested through experimentation or through additional observations. And that's what typifies a good hypothesis. Not a guess because of a hunch that we have, but well thought out explanations that can somehow be tested. And so what I thought we would do, rather than go through each step of the scientific method, I thought I would present you with a case study, one that you might actually have heard about before, and really analyze this case study to look at how and why hypotheses were proposed, and look at the experiment that was performed, look at the data that were collected, and talk about why this is an excellent case study to understand this process of scientific inquiry. So, what's the case study? We're looking at coat coloration in old field mice. So that's what's pictured here. You see two different old field mice that are part of the same species, but very obviously they have different coat coloration. Now, as lovers of animals, I'm sure you've observed similar patterns before. Animals, even within the same species, can have very different coloration patterns. It can range, like these mice, from very dark to very light, or it can be a little bit more subtle than that. If we compare different species, we also see that there are clear differences amongst species when it comes to coloration. Now, why? Why is this, right? This is our observation. There are mice and they are different colors, even though they're part of the same species. Why? Why are they different colors? Well, we're not the first person to ask why. So, Francis Bertodi Sumner was a scientist back 100 years ago who, made some observations Francis Bertodi Sumner was a scientist who 100 years ago made some observations about this species of mice, Paramiscus pollinotus. These old field mice are found in a lot of different places in the southeastern United States, including Florida. And he observed them in these two different areas. We see a shore-based beach population and a population that's a bit more inland in the Florida Panhandle that has a slightly different ecology. And he noticed that even though these two populations were the same species of mice, they had these very different color patterns associated with them. And so he thought to himself, what would specifically drive such evolution? So I want you to think about this. If there were two different species of mice in two different areas that were two very different colors, what explanations might you propose as to why there are these two different colored populations? Now, there's a lot of ways you could go about thinking about this. You could do some research. You could look into other animals that express different coat colors by going to the scientific literature. You could look at other populations in the area to determine if they also differ in their colors. Or you could just relate this back to, the knowledge that you have of ecology and evolution. What might drive this? So think about that for a few minutes. Now, I can come up with a few ideas. If we think about animals, we know that diet has a significant effect on coat color. Think about flamingos. That's a great example. We know that the pink coloration of flamingos is because of the food that they eat. Maybe coloration has something to do with the food that these mice are eating. Okay, I know what you're thinking. You're like, no. The first thought many of you probably had was it has to do with camouflage, right? That these are two very different habitats. These two different habitats are very different in their coloration, and so maybe the mice are trying to blend in. They're trying to blend into their coloration. Now, why are they trying to blend in? To avoid predation, right? You would think that they have different predators, maybe bird predators, maybe some larger mammal predators, a fox that might want to eat them, hawks that might want to eat them. So maybe they're trying to blend in. And that's actually what Sumner proposed. In his 1920s paper that he published in a small journal, he said that he thought that these two populations evolved this way because it allowed for better camouflage, so they were better protected from predation. But he didn't actually do any sort of experiment to determine if that was true. He didn't make any sort of observations. He just proposed this hypothesis. So this leads us to this process of science. We can propose an idea, but how do we test that idea? Good hypotheses, good research questions lead to good predictions. We can make a prediction. If the variation in coloration has to do with being better camouflaged in each environment, then those mice that are not camouflaged in a specific environment will be more likely to be preyed upon than mice that are camouflaged. That makes sense, right? If coloration protects from predation, then mice that don't have that specific camouflaged coloration, they would be more likely to be preyed upon. They would be selected against. So we don't have to do that experiment because someone already did. Dr. Hoekstra and her students in 2010 decided that they would look at Sumner's hypothesis and actually propose an experiment that would test this hypothesis. So she and her students did this really interesting experiment using artificial mice. So a whole bunch of undergraduate researchers took clay and paint and made these artificial mice, and that's what's shown in this picture here. We have light mice that are made out of clay, and we have dark mice that are made out of clay. So these models that they used weren't about to run away, right? That was sort of a key point of the study that they were doing. They couldn't run away, but to a predator, at first glance, they look real, right? To me, at first glance, they look like they could be real mice. But these are artificial mice that are just made out of clay, some that are light and some that are dark, matching the populations that normally exist in this Florida, and these two Florida populations. Now, what did they do with those mice? They actually took both types of mice, the artificial light mice and the artificial dark mice, and they put equal numbers at both field sites. So at the beach field site, that looks kind of light, where the light population is normally found, and then the dark inland area, which is where the dark mice are normally found. Now, what's important is, like I said, equal numbers of both types were placed at both field sites. So light and dark mice are put into the beach area, the lighter area, and light and dark mice are put into the inland area, the darker area. The scientists and Dr. Hoekstra and the undergraduate researchers, they put out the mice, and then they came back the next morning to retrieve their artificial mice. And they counted how many models were missing or how many models were damaged. So they looked for bite marks or claw marks, or if a mouse was completely missing, they assumed that it was preyed upon, that someone took it away because, these model mice couldn't walk away by themselves. And so they counted those all as attacks on these mice. Now, let's think about this experiment before we get into the results. This is an excellent controlled experiment. Controls are the types of variables that scientists use in order to make comparisons. Controlled experiments compare experimental groups and controlled groups. In this specific case study that we're looking at, the experimental group are those mice that were non-camouflaged. So the mice that were white in the dark area or dark on the beach. Those would be the experimental group mice. The control group mice were camouflaged. So they were like the normal population that naturally inhabited these two areas. So they were the dark mice in the inland habitat and the light mice in the beach habitat. Now, ideally, only the variable of interest, the one that we are trying to collect, the effect of coloration on the behavior of predators, is what differs between the two environments. So that is why the scientists used both types of mice in both areas, so that they had control and experimental groups in each of the two environments that they were looking at. Having controls means that we can ignore or cancel the effects of unwanted variables. Now, it doesn't mean that everything is held constant. Obviously, the beach is a little bit different than the inland area, right? We can explain lots and lots of differences. But it just means, a controlled experiment just means that we're trying our best to keep everything equivalent at the sites as best we can in order to try and focus mainly on the one variable we're interested in. So it's not like these scientists decided they would change the size of the mice in the different areas. They didn't do this on different days, right? They didn't test the beach one summer and then the inland area in the winter, right? Because that wouldn't be holding things consistent. So good controlled experiments do their best to be consistent and collect the data in a consistent way as best they can. It's not always possible to hold everything constant, but scientists do their best. Now, speaking of that term variables, we talked about holding things constant, holding variables constant. Variables is just a term that we use to talk about factors that can change within our experiments. And we focus on independent and dependent variables. We'll talk more about these in the upcoming weeks. But very briefly, independent variables are the variables that the researchers manipulate. So independent are controlled by me. I control them. The independent, I control them. I control the independent variables. The dependent variables are what is measured within the experiment. They are the data that we collect. The data we collect are the dependent variables. So think about this experiment. Think about the independent variables and the dependent variables. What variables were controlled by Hoekstra and her students? What data did they collect? So identify those and see if you agree with what I'm saying. So the independent variables in this experiment are the camouflagedness of the mice within their given environments. Is camouflagedness a word? I don't think so. But we controlled whether or not, or I should say Hoekstra and her students controlled whether or not the mice were camouflaged, were very similar in color to their environments in both the beach and inland environments. And they controlled the mice that were not camouflaged, right? So they did not blend in the beach and in the inland environment. Now, what was the dependent variable? Did you write down that it's the number of attacks or percentage of attacks? That's the data that was collected. Those data were collected by looking at bite marks or claw marks, or if the mice models were missing. So they collected that data. That was the variable that was measured within the experiment. And the dependent variable is what we think will vary based upon the independent variable. So we think that the dependent variable is going to change when we look at the control and the experimental groups. So our different levels of camouflagedness, if they are camouflaged or not. We think that that is going to influence the dependent variable, the data we are collecting. So I was tempted to just do an analysis of that experiment without actually giving you the data, since I'm very interested in looking at the experimental design and how it relates to our hypothesis. But I always need to know the data, right? I always want to know the data. Show me the data. So here are the data that were collected. You can see that, again, they were comparing the beach and the inland habitats, and they were looking at the camouflagedness. So we have our light mice camouflaged in the beach, and we have our dark mice that are camouflaged in the inland population area. And then we have our dark mice on the light beach and our light mice in the dark inland scrubby forest area. And then we have along our y-axis our dependent variable, and that is the percentage of attacked models. So of all the models that were put out, what percentage of them were attacked? And you see this lovely pattern of behavior. Now, I don't have the statistical analysis in front of me, although maybe we should read the original paper, and then we would get an idea of the actual analysis that they did. I'll pull that up for our discussions on analysis. But you can see here a clear pattern where the light models were attacked very rarely in the light environment, right? About 25% of the time. Similarly, in the dark environment, the dark models were attacked about 25% of the time. So not zero, not zero. They were attacked a bit. Of course, right? The predators are able to identify their food products even when they're camouflaged. But those levels are much, much less than what we see in the non-camouflaged groups. In our dark mice group on the light beach, you see about 75% of those were attacked. And with our light models in the dark inland habitat, you can see again about 75% of those models are attacked. So what does this show us, right? This surely has to do with predation. This camouflaged color, right, does protect these mice that blend in with their environment on some level compared to the non-camouflaged mice. And so this directly relates back to our original hypothesis, our original predictions that the coloration that we see different between these two populations has some sort of tie to the predation levels, right? That this coloration has something to do with being camouflaged, blending into the environment. So why did I choose this specific study for us to focus on? One, like I said, it may have been something that you discussed in previous courses. It also was easy in the sense that being camouflaged, right, is something that you're familiar with if you've ever thought about animals before or studied animals before. But why else? Why did I pick this specifically for this research methods class? Well, one, a clear hypothesis with a prediction was tested, right? Clearly we were looking at how the coloration was related to the level of predation. So this was a very clear hypothesis with a very clear prediction. This led to a very elegant, well-controlled experimental design. Elegant is a term that we use often when describing experiments that are, to be quite honest, a little bit simple. And I mean that in the best possible way. I know simple is not often a positive term when discussing science, but actually simple, straightforward experiments are the best experiments that we can do. And that should always be our goal. We should always design simple, well-controlled experiments. Those simple experiments could be part of a much larger, more complex, interesting study system. However, each individual experiment should be as straightforward, as well-controlled as we can make it. And that is what Hoekstra and her students did. They suggested this simple experimental design, this elegant study, that led them to clear independent and dependent variables, right? Very obvious indicators of being camouflaged or not. Very clear data that were being collected, measures of predation, that allows for the support or rejection of the hypothesis. They knew before they did this experiment that whatever data they collected would clearly support or reject the hypothesis. Either being camouflaged helped these mice or it didn't when it came to predation. Either this coloration was directly related to how often they were being preyed upon, or it wasn't related at all, right? There was going to be an answer when they collected their data. And that's our goal. That's our goal in this class. That should be our goal as scientists in the future. We should be thinking about studies that have clear hypotheses, that we can have elegant, well-controlled experiments that collect obvious data, and those data should go back to our predictions, should go back to our hypothesis and clearly support them or reject them depending on the outcome. Now in this case, Hoekstra's data did support the hypothesis, but that's not what makes it a good study. If Hoekstra's data had rejected her hypothesis, if there were equivalent numbers of predatory attacks on the different colored mice in the different environments, this would still be a good study. So it's not just that, like, yay, she won, her hypothesis was supported. It's not that. That's not what we mean by a good scientific study. What a good scientific study, its results are clear and obvious, and they will support or reject that hypothesis. Rejection of a hypothesis, even though it may not seem like it, is just as valid as supporting a hypothesis. And so that's going to be our goal. Our goal is to go through this process of scientific research where we're asking interesting research questions, where we're spending time within the primary literature looking up the experiments, the observations that other scientists have already published about. Then based on our question and our background research, we are going to come up with elegant hypotheses that try to focus on one aspect of something that we are interested in. We are not going to be able to explain the entire field of animal behavior as related to one question. Little questions asked again and again get us to big ideas and theories. Once we have come up with an elegant hypothesis that will lead us to an elegant experiment. Now, this experiment could be something that is done within the laboratory with proper controls, or it could be observational. So I don't want you to think that this has to be a laboratory-based experiment. Hoekstra did a great job of doing some experimental manipulations within the field, but even that is not always possible. Sometimes field work has to just be observational, but we can still test hypotheses even without having strict experimental groups. And the goal is always to design an experiment that, regardless of our answers, regardless of the data we collect, they will somehow relate back to our hypothesis. Now, how are we going to know? Well, we have to analyze the data that we collect. We need to draw conclusions from our results. So this has to do with looking at statistical analyses that we will talk about. And then again, we relate that back to our hypothesis. Whether or not our hypothesis is supported or rejected, we are always entering back into this loop. We are reporting our results so that others can see it, and we're reconstructing our hypotheses to ask more and more questions. So I challenge us in this class to come up with these elegant experiments like Hoekstra did so that we can answer some really interesting research questions. So with that in mind, you should be working on article summary number 2. That is going to be due by next Tuesday. As we talked about in class, the remaining article summaries will be based on articles that you search for and you find within the primary literature. So you should be looking for a research article now. You should use all those search engines that we talked about. If you're having difficulties, of course, you can contact me at any time so that we can find you an article. The article should in some way be related to something that you're interested in for your research proposal. So it could be a paper that's about the species you're interested in or the concept you're interested in. But make sure you're looking for a primary literature research article that you can read and summarize by next week. So make sure that you're doing that. As you're doing your search, you may also download other articles, you might want to do that, since you will be continuing to look at these article summaries week after week that all relate to your research proposal. So make sure you're working on that, again, due by next Tuesday. This week's knowledge celebration, due by Thursday at 9 p.m., is actually a literature scavenger hunt. So this is going to help you as you are diving into that scientific literature and using all these search engines and using the Canisius College Library website. That is posted on D2L, as I told you. You can type right into that and re-upload it to the Dropbox on D2L. Again, that is due by Thursday. The other thing I want to mention is that next week, on September 24th, we don't normally meet on Thursdays, but for the next few weeks, I wanted to give you the opportunity to meet some other scientists in various fields. So we'll talk about scientific studies that these scientists have been working on that relate in some way or another to animal behavior. Of course, it has to be virtual right now, so we're going to do a live Zoom session. So starting next week, the 24th, please mark your calendars, all of us, whether we are online only or we meet in the classroom on Tuesdays, on Thursday, please mark your calendars. We will have a live Zoom session at 11:30 where we'll all sign on to Zoom. I'll send you the link for that. And we will have a special guest speaker. So I'll remind you next week, but I wanted to make sure this was on your calendar. It's on the syllabus, but please make sure you have it marked for next week so that you can come and join this conversation. It'll be somewhat casual. We'll have plenty of opportunity to chat with our scientists. So please, again, mark that on your calendar. I think you'll really enjoy it. So contact me if you have any questions, and please review this video and take notes and start looking for those article summaries, or looking for those articles so that you can get your summary started for next week. Hope you all have a great day.